Today, this is the last session of 1 week online international e-FDP and e-STP, empowering pharmacy education, navigating regulation, global trends, and advancing outcome-based education and research. This is the last session. Uh, I would like to introduce Dr. Om Prakash Katare, sir, the eminent speaker from our pharmacy field. Sir is going to talk on the topic of industry academia translational research, a case study on at on the Punjab University campus and beyond. I am feel pleasure to introduce Dr. Katare, sir. Sir completed their doctorate from Hari Singh Gaur Vishwavidyalaya, Sagar MP in 1992 and graduation and post-graduation in 1981 and 1983 respectively. Sir has a huge experience. Currently, sir working as a Director Research Promotion Cell, Punjab University, Chandigarh from last 2 years. Dean of Faculty of Pharmacy Science, Punjab University, Chandigarh since 1 year. Department of Head University Institute of Pharmaceutical Science, Punjab University, Chandigarh. Sir is a professional and academic member of All India Board of Pharmacy Education and All India Council of Technical Education, AICT, New Delhi, since 2 years. Sir has a huge experience on various advisory committee. I will list here few. The Chairman, Institutional Animal Ethics Committee, Punjab University, Chandigarh, since 2017. Member of Advisory Committee, AICT Syllabus Committee since 2018. Member of Institutional Ethics Committee of Postgraduate Institute of Medical Education and Research, Chandigarh since 2017. Sir is a member of Project Department of Biotechnology Task Force since 2005. Sir is also member of Selection Panel Council for Scientific and Industrial Research, University Grant Commission, Department of Science and Technology, Indian Council of Medical Research. Sir is expert on Scientific Board International Phospholipid Research Center, Heidelberg, Germany since 2006. Sir has a huge academic experience since 1991 to till date working as a professor. Sir grabbed many awards in his career. I will list here few. Sir is a president and Deputy Tech Transfer National Award 2007 by Government of India. APTI Best Pharmaceutical Scientist Award in Apticon 2017. Best Patent Award in 2012. Sir is figured as world ranking of top 2% scientist in the Indian subcontinent issued by the Stanford University, USA, and many more. This is an overview of Sir research publications having more around 15,000 of citation with H index of 68, and Sir has evaluated more than 50 postgraduate research having 20 patents, more than 290 publications, edited 5 books and 21 book chapters, and more than 150 expert talks is delivered by the Sir. I will request Sir to please start the session.
Thank you so much, Dr. Prasant and team. I hope I am audible enough. मेरी आवाज आ रही है?
Over to you, sir.
Again, I repeat, thank you so much, Dr. Prasant and team. Am I audible?
Yes, yes, yes.
Am I sufficiently audible?
Yes, sir. Yes, sir. You are audible, sir. Over to you.
इको हो रहा है क्या? Is there any echo?
No, sir.
Okay. So, expressing my pleasure and uh, the joy to be here virtually, uh, Chandigarh to Nasik. है ना? Thanks to the technology, we are able to meet virtually. I don't know why the audience, our fellow teachers, are so much eager to listen me. But I am really eager to interact. So uh, with this, uh, joy, I am happy to present the title that is chosen is Academia, the breeding ground for research and innovation. How to build the ecosystem is a trillion million dollar question. It is obvious that in uh, uh, in foreign countries, universities, this is a norm. This is not something new that we are imagining here in India. That our educational institutions should be capable enough, should be equipped enough that we should be able to become that the nursery of the researchers, nursery of the research and innovation. But there are many questions. Uh, so in my presentation, I would like to take a case study and then addressing the possible probable questions, how to do it. So the Punjab University, you all know, this is our campus, iconic building, Gandhi Bhavan. Uh, visited Punjab University campus, the oldest university in this area. This is actually the university which migrated from Pakistan. The University of Punjab is still there. So it is around 1884 somewhere. It was its inception. Then after partition, it came to Chandigarh through Amritsar, Solan, and Shimla. So this is a very old heritage status university, the Punjab University. So with this brief, the academic setting at Chandigarh, I move forward. May I have a second slide? The very first thing to set the ball rolling, सबसे पहली बात यह है कि be on academic platform and otherwise also we should have this the attitude of being खोजी. हमारे मन में एक खोजी भाव होना चाहिए. हमारा मनोभाव जो है, एटीट्यूड जो है, वो एक खोजी का होना चाहिए. एंड खोजी कोई छोटा मोटा नहीं, थोड़ा सा बड़ा सा खोजी होना चाहिए. वी शुड डिफाइन अवर गोल and what else be on a applied side in pharma. The basic sciences we cannot expect, but we are on the applied side, so it is our fundamental duty to have this translational work, not only giving the publications, the anything which is remain in the lab is not usable. So the research should come out of the lab and reach to the its real the practical application. So it's very important to understand uh, the importance of the attitude. How do you think? What is your mindset? So making mindset, how to make a mindset is also a question. One question leads to another question. So researchers quest the खोजी बने. So when we talk of this, the goal, we must take this the importance of the question that the gravity man, the Newton, we see in the picture, the importance of the question. हमें प्रश्न करना आना चाहिए. अगर हमें प्रश्न करना नहीं आता है, वही हम फेल होते हैं. So this man, this gravity man is teaching us that we should ask big question, bigger question. That this apple was falling down, but only the scientist, that time it was declared fool. एक ही लोग उसको बेवकूफ कह रहे थे कि ये बेवकूफ है. But before asking this question, he had the answer in mind. And the importance of this picture reveals the importance of his mindset. What sort of a mindset he had? How did he develop the relaxed mind? So this is a very important thing that we should ask bigger questions. Next. Now with this mindset, I would like to come to the story that University Institute of Pharmaceutical Sciences. It is visible in this slide that we have the role model, the father figure in pharmaceutical sciences, late Padma Shri Professor Harkishan Singh. This department is known by his name, by his research, chandonium iodide. And in this, the atmosphere, the ecosystem was according to this image. So I joined this department in 1997. And I had this manobhav attitude of being khoji and thinking something that justify my presence in academia, pharma academia, that I have to do beyond publication. And like Professor Singh had shown that time, that a molecule, the only molecule given to the country from the academia side, it's a rare thing. Still we celebrate this achievement of the UIPS. So you can just imagine the kind of a ambience, the kind of a vibratory environment is prevailing that time when I joined the department, and I had these aspirations to work with. Next. So in medico pharma research, we are actually facing this problem that the synthetic molecules, the molecules, the drugs, mostly coming out of our synthetic labs, they are actually very toxic. So Mahatma Gandhi had this statement he criticized. He was very critical. He was blatantly critical. I would say that he would like to die, but not to take chemicals, because they not to take medicines, because they are the chemicals, and they can actually do lot of harms without any experimentation. The science in the lab, he had this clear cut observation that these molecules which are which can disturb the harmony within the biological system, the system biology, they can destroy. So Mahatma Gandhi gave us a challenge that how to overcome these problems, that our drugs are not safe, and even if they are effective, and compliance is also compromised. So our researchers should think of it कि हम ऐसा क्या करें कि जो हमारा फार्मा है, जो ड्रग मॉलिक्यूल्स हैं, उनको हम कैसे safe. And people are doing and achieving some success also, but yet we have a long way to go to cover the journey. Next. So I will quickly go through to cut the time, because we have to cover up the safety issues as you see in the picture. Jump, bump, and dump model. When the drug is taken, it actually takes its own journey, sojourn, to go here and there. There is no control over the kinetics, the distribution, and dynamic. The pharmacodynamic is also accordingly not up to the mark, or misdirected. So we need targeting, we need delivery, we need formulation approach, we need so this to work with the molecules, whether at the chemical level, structural level, and extra structural level through pharmaceutics. So from dosage form, conventional dosage form, to drug delivery. That's the idea of my work. And that, please, next. So in search of an ideal career, what to do? We have to have the drug molecules in such a state of entrapment or encapsulation that it should go right, avoiding the unwanted these site locations, and then on a right track it must stay on, and then it should act on the target side very specifically. If this happens, we will be able to transform the behavior of the old drugs, giving them a new lease of life. एक नए अवतार की possibility है. If we are able to play with these excipients, the methods, the fundamental principles of the drug delivery and nanotechnology, we can do it. Next. So liposomes, by now everybody is aware what they are. When I started working for my PhD, it was a little known entity, and people were even not believing if there is any such type of structural creation there. So now there are a lot of interest from all over the places, all over the sciences, scientific field, interdisciplinary. It has become now, and now pharmaceutics is not the only one, but pharmacy is not the only one, but from every other field, the people are taking interest, and they are exploring the potential of liposomes, how they can exploit it for the varied applications, not only in pharma, but in agriculture, in diagnostics, in gene delivery, even in agriculture. Next. So the basic building block of liposomes, this thanks to this nature given molecule, the phospholipid, we all know that we are made up of all these membranes, the phospholipid membranes. Our life is because of this molecule, that cell membranes are formed in such a way that they can host the biology within. So this phospholipid molecule has this unique tubular structure with two phospholipid chains and a polar head group, which is linked to the glycerol backbone. And because of this beauty in phospholipid macromolecule, they can join the other molecules of the phospholipids, making a polymer kind of a arrangement, polymeric arrangement, with the use of the non-covalent bonding, and they can make the thermodynamically stable structure, which can become the host, the carrier, the delivery system. So the water is very important. It is not the phospholipid. The important, the people do not give the attention to the water. Thanks to the water, its ability to create this intramolecular interactions that leads to the formation of the supramolecular assembly, the self-assemblies, they self-assemble, and during this process of self-assembling these molecules, they can encapsulate, they can entrap and the molecules, very small molecule to the large, very large one. The variety is there depending on the composition, depending on the method, depending on the selection of the various environmental factors like temperature, pH, presence of electrolytes, etc. Next. So they are unique in their characteristics and their ability to entrap, solubility, their targeting potential, their compliance, their functionality. It's the diversity. It is the diversity and the flexibility that they provide. They stand apart. Unlike any other carrier systems, they provide the broad spectrum possibility all around, and they can be tailored according to the need, the medical application, and the need of the molecule, the drug, the situation. So they are of very high interest for all. So variety is there. They can, as I said, आपके पास में बहुत सारी possibilities हैं, उनको हम create कर सकते हैं. Not only the vesicle system, they can create the micellar system, emulsified system, complex membrane system, liquid membrane system. बहुत सारा इसमें हम उसको create कर सकते हैं. It is you can understand the in pharmaceutics there's always the scope of using another component to combine with and to create something new, something very new. And if you find novelty there, the patentability is there. Next. So the technology features are also different than conventional system. We have to have this understanding of these things: the phase transition phenomena, the critical packing parameter, the micromagnetics, surface modifications, and all the instruments we use in the technology. Next. So these systems are characterized differently. We have to have, unlike the tablets and capsules, we have to have the different parameters. इनकी हमें जो information है that into the of their structure is taken differently. For example, we may need to know the lamellarity, the number of layers, lamellae, its a change in the phase. The changes from one phase to another, entrapment, loading, release, leakage. The concentration of the excipient itself is very important, and the stability is very, very important when they are prone to hydrolysis and oxidation. Next. So we have liposomes in market. That was encouraging the proof of the concept when in 1964 they were first seen under the microscope, under the transmission electron microscope. It was just in the hands. They were just in the labs of the biochemist and biophysicist until 1971 when the Gregorius got this application, medical application for I think it was for the liver. Then since then, for a long time, the liposomes were struggling to find the way to get into the clinic. It took a very, very long time because of the leakage problem and stability problem, and because they were quickly cleared by the RES system of the this clearance, elimination from the biological system face problem, but thanks to the cholesterol that we could produce the stable liposomes, and this translated, and they are there in the market. So very, very encouraging this historical this success at that point of time. Next. So in my work, this is the case study down the lane in my 27, 28 years of the past history at Punjab University. I have this, you just had it, that number of publications, 35, around 35 PhD, so many patents. But this story, the old story, of course, but it is still a gold standard story, which reflects on how to approach. So I always like to present this story because I could not find any other to substitute this thrilling journey which actually was the journey where I begin, and still it gives me a lot of this the memory, the thrilling memory that I enjoy today, even today, while there is so much to do being done. But this diethanol, the drug, the disease, the psoriasis, the dosage form, the liposomes, the three things which we have to consider for the new researchers. We have to focus on the possibilities, what to do, where to go, and how to begin. Next. So I will not go into detail. Psoriasis was the disease, candidate disease, where we have the hyperproliferative skin, and because of this, there is a lot of pathophysiological changes, which is very, very difficult to tolerate. And we have this 130 million population worldwide affected. We have 75 psoriasis association, and there is association for association. One can understand the gravity of the problem, the severity of the problem. So we have systemic therapy and topical therapy. Systemic therapy, the drugs which are used are quite problematic, and topical therapy like corticosteroid is also problematic, and sometimes not able to be to show that effect. So both routes, both approach are problematic. So there is a scope to work to give something better. So with this understanding, I started working on this molecule, diethanol. Next. So the diethanol anthralin molecule is a very good molecule, but its irritancy, burning, and staining, erythema, they are all overweighing, overburdening, and make it a fearful drug because of its free radical formation, and its efficacy is good, but this side you can see that because of this non-compliance region, this has become the non-usable. It has good action, therapeutic mechanisms at the cellular, intracellular level. Ubiquitously, it hits upon the targets. So, by virtue of its mechanism of action, it is an extremely good molecule, but extremely bad molecule. So, this paradoxical situation we understand, we face this in this. So, this is the challenge actually, how to manage this while retaining its goodness and eliminating its this bad temperament. So actually this idea of working on diethanol molecule was given to me by thanks to this PGI, the medical institution. The head of the department of dermatology, Professor Bhushan Kumar, is just for me, he was the great mentor. Because of him, I could embark upon this journey, and right from the beginning, the first PhD thesis, we started working with the medical fraternity. This is very important in industry academia interaction, that the role of the medical institution, the clinical partner, is very, very important. Because it matters a lot when we are looking for the innovation, the new thing. So in the current scenario, our industries are not actually uh, very much interested in creating something new, but they are only interested in generics. We are number so and so, number 3 and all. We are celebrating the success, but actually we are not working for the new things. So this experiment was actually at that point of time was a very new thing, that you start working with the clinicians, and then industry came later on. So he told that this drug, that time there was a product like US Betamin product that was not satisfactory, and patient in compliance was there. So the problem was given to me, and we started working. So the German researcher Mustakelio actually gave this landmark statement that he wrote in journal in 1992 that I doubt whether an entirely non-irritating diethanol derivative or treatment modality will ever be developed because there is something in the molecules which need irritancy to work with. So in desperation, in हताश होके उसने ये statement दिया, and this became the challenge for us to overcome and to remove his desperation. We started working. Next. So the conventional approach so far with this molecule has been the conventional. The attempts were made to modify the vehicles and some clinical modalities, some other methods to use occlusive coverings after application of this diethanol to enhance its penetration so that it can work well. So these conventional approaches did not work well, and the dermatologists were actually struggling to find something to overcome non-compliance. Next. So what is the paradox? What is this opposing this behavior of diethanol? The drug is actually undergoing keto-enol tautomerism, and this gives the free radical, enthronal radical, and then it gives the second generation these radicals. So by virtue of its faster track degradation, the desired middle enthronal radical is not available actually for action, but rather enthralin, this brown, the undesired, not desired, this which is responsible for the staining and irritancy, it goes down and it creates problems. So you see in this mechanism, we were the first to identify this mechanism, the problem that we have to work with this problem, the fast track degradation of diethanol. Next. So in search of solution, the brain is storming and mental threshing. People ask what is there for academia in research. This idea was generated in the classroom situation when I was teaching pharmaceutics, the physical pharmacy, where I encountered this idea that the lecithin, it is a known antioxidant capable to slow down the degradation, should be used in such a manner that it can be used for these diethanol. So that it clicked during the one classroom. So it's a classroom idea, academic idea, actual academic idea that it that time thrashed on. Next. So this is the idea out of the box, and we did this the first PhD thesis that the real life picture of this entrapped drug within the multilamellar vesicles made up of phospholipid and the combination of the various the other excipients we used. So it modified the reactivity and it moderated the reactivity degradation. Henceforth, we got the mechanism of action of diethanol according to our favor in favor of us. Next. So this is how we created novelty and the inventiveness within the frame of this research work, and we got the strategically placed drug molecules in and out, so that we can create the innovation where we get some molecules quickly available and some are on a controlled rate. Because of this idea only we got it patented worldwide, and we filed PCT with the help of the company. Otherwise it was not possible to face the patent, because by then the liposomes were the known art, and it is not easy to patent it, because it is assumed to be obvious, section 3D. So we had this thinking, so we could face the patent. Next. So we did the clinical trial, the academic clinical trial, without even spending even a penny. We had this clinical trial on 21 patients in PGI with the help of their senior resident, Dr. Saraswat. He is the leading dermatologist in Lucknow. Yet still he loves this product, and however this product could not reach to that level of marketing, but it is a very because of the marketing muscle, the company could not market it, but this clinical trial that it worked at lower doses, the same as that of higher doses of the marketed preparation of US vitamin. So this was a heightened joy, the enthusiasm it created at that time to see the results in clinic after this on our rat models. This is something at that time, a lot of celebration for this. Next. So Life Care came into the picture, and we got this in collaboration. Next. Next, please. So we got the technology transfer on 2nd May 2006. So on campus, lot of celebration. काफी इसको highlight किया गया, और this was supposed to be the this kind of a tech transfer, the first technology transfer of this kind on campus. So there was a lot of appreciation, recognition, laurels, applause all over the campus, the Tri-City, that we had with the Life Care innovations. You can see in the picture our vice chancellor, our dean, and our in his specs, Professor Bhushan Kumar and Dr. Verma. So this tech transfer celebration it had. Next. Then post tech transfer issues were really very challenging, because the liposomes were not easy to make, and we had all sorts of this issues scaling up standard operating procedures. Their design of experiments and all these were done with the help of our colleague Professor Bhoop, the master of QVD. So we collaborated with so many people, and then we got it from lab to industry, covering all these steps. This post lab work was more than that what we did in the lab. So the material and all the costly material, the monitoring, the reproducibility, and all sorts of things. It was a really difficult to find the way for its commercial level production. Next. So we had the product finally turned into the reality. The concept, the academic concept, from concept to commercialization, from lab to clinic, zone. And this is how they marketed. Next. So story goes on to be did for the next product. We did for coal tar, this time for nanotechnology driven. This is a different approach where we used less costly material, and we could give the product at affordable cost. This has really been working very good, but again, a marketing muscle could not match our aspirations. This actually market बहुत अच्छा नहीं हो पाया. Next. We did so many other research that some examples that we could entrap the bacteriophage within the lamellar system, and with the help of the microbiology professors, we could produce something, the novel idea how to entrap this bacteriophage for the to overcome the biofilm, the resistance. We got 2 patents, and the ICMR, thanks to ICMR, the work still is going on, and we hope that it will be on the translational path. Next. So another one for the methotrexate delivery, please. Next. Next, please. Now, so problems are plenty. Not this is just a glimpse, just a spark kind of a thing. We have to work a lot while we have not yet done much. There is a long way to go. बहुत कुछ करना है अभी. Problems like cancer and the diabetes, not to name, but everyone will know the pharma is actually having a lot of work. Our medical fraternity is very good, but they are looking to us for the actual solutions. Who will give this research? So we have to go beyond our own ivory towers, and we have to collaborate. We have to join hands. So we did it. Industry and the various institution networking, collaborations, joint ventures. This is the need of the hour. Next. So I will not go into details of the academia mindset, industry mindset. They are the 2 different mindset. We have to understand the priorities, the strength, the gaps. जितनी कमियां हैं, खामियां हैं, हमें अपनी समझनी पड़ेंगी, and industry का mindset भी समझना पड़ेगा, so that we can bridge the gap. This all depends on your aspirations. अगर आपके aspirations high हैं, आपका एटीट्यूड वैसा है, और आपके अंदर वो है, जज्बा है, जुझारूपन है, तो आप कर सकते हैं. नेक्स्ट. नेक्स्ट प्लीज. May I have a next slide? Next, please. So how to make it possible? Now, the million dollar question कि इसको हम करें कैसे? How to do, how to make it possible? ये जो लिमिटेशंस है हमारा, वो क्या है जिसकी वजह से हम कर नहीं पा रहे हैं? Why we are not able to come up to the expectations, societal expectations, the medical expectations, our own expectations. So we have the history of in Indian history, the ancient Indian history, that the example, the संजीवनी, तो वो संजीवनी हम लेके कैसे आए? ये सबसे बड़ा क्वेश्चन हमारे दिमाग में होना चाहिए. अगर नहीं है तो होना चाहिए. तो जब तक हमारे उसमें एस्पिरेशंस नहीं होगी, हमारी आकांक्षा नहीं होगी, डिजायर नहीं होगी, तब तक हम आगे नहीं बढ़ सकते. सो हाउ टू मेक इट पॉसिबल? दिस इज ए वेरी फंडामेंटल, क्रिटिकल, mind thrashing question. Next. So search for the master key. What matters most? Next, please. May I have next slide? 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 Something goes wrong. You can scroll. Yes, please. हाँ. इसके पहले वाली स्लाइड, before, bring back. Yes. So this product, this National Technology Award, and I had the fortune. I was fortunate to shake hands with the Kalam Saab for this Sorizome success story, and it could and beside this shaking hands, shaking divine hands rather, I had the 1 lakh cash prize also. 1 लाख रुपए भी दिया कलाम साहब ने. And next, please. An OPPI Scientist Award. This international recognition came that Phospholipid Research Center, Germany. I was appointed on board as a founder member. When they took this initiative of Phospholipid Research Center in Germany. So I've been this place, Heidelberg, and so this was the international work, translational work. Next, please. OPPI Scientist Industry Award. This was given in 2011 at Hotel Taj, Mumbai, when this Lipotar was launched. And again, I received 1 lakh cash prize for this award. Next, please. And there's a lot of recognition, not only for me, but for our students also. So, international student this award for Dr. Kesar Raza, and this has been our Professor Bhupinder Singh Bhoop. So there are many awards to our students, not only to Dr. Kesar Raza, but many won the high-end these awards and the laurels. Next, please. So this is the recent success with IPCA Labs. We could launch this product, and we are doing fairly well in the market, because this time the marketing muscle I found. So the initial success could not reach to the market, but हमने हार नहीं मानी और हम आगे काम करते रहे. So with thanks to this collaboration with the IPCA Labs, we could launch this product, and we had a lot of struggle in getting this product into the market. We had to overcome the mindset, the marketing mindset, because they were not ready to experiment with the new things. They were happy with the old things. So a lot of negotiation, and we had, and finally we could win their mind, and we could launch this product, Itodolac gel, and it is doing fairly well. Next. Another milestone that we had the other product, the Folitrax LP gel, with the help of the company Alina, of course for IPCA Labs only. In this product, we had a lot of problems faced with the scale up, but finally we overcome and got the product translated. So this is our team, our research team group at the time of the tech transfer. So there was a real life joy to see the product, the lab product, converting into the commercial scale. This on the shop floor. So nothing like it when students see their imagination. You can imagine their enthusiasm, the spirit, the joy they would have when they see such things, and this improves. This actually gives them an edge over others when they have this kind of a entrepreneurial approach, this industriousness. This is the demand of the day, that we should prepare our students, we should expose them to such kind of a translational things, not only to the research to be folded within the papers, within the labs, within the documents, but it should come into the real life practice. So again, so this is something unique in the something different in the country scenario, and out of that, university receives a return also, and royalty back to the university. The revenue to the university is something rare phenomena in the Indian scenario. But by the Almighty grace, we could demonstrate, though it's not a big thing, we feel अभी बहुत कुछ करना है, लेकिन ये हो सका. Next. So some awards that this is the other part. Actually, my presentation is somehow distorted. Otherwise, I wanted to show something about the youth empowerment activity, the self-development, the personality development programs we are carrying on campus, which I cherish more than that of my research nowadays. So we have been working with youth, and especially the heartfulness way. So I represent also this meditation practice, practicing and training institute, the heartfulness, where we won this award, which I relish more than our Rashtrapati President Award and other awards given to the Punjab University. We are receiving on behalf myself and my colleague professor, and this meditation hall, which can host around 1 lakh the meditators, the first in the world. And this is something which I really wanted to share with the college people, that this is the other part which is very, very important to take up. Until unless we work in this area where the of the mind, the capacity, the personality, the character formation, then only we can achieve this the goals of research, the entrepreneurship. So this is the holistic approach that institute has to adopt. The research will flourish like anything if we work on this fundamental bottom line of the our, it's a capacity building program rather. So this heartfulness way, we have the lot many activities going on campus Punjab University. We have a structured program, modules, faculty development program. If institute wants, I can facilitate this program. Many colleges are doing this program in the Yamuna Nagar and ISF Moga, and they have adopted in SKV Nagpur. So they picked up this program, and I was the one who introduced this idea, and they are really reaping good the benefit in a, so where we have to work at 360 degree. So though it is called personality development program, but it is all about the inner persona, the character formation, the capacity of the mind, the focus, the concentration. This technique is very useful, we have found. Next. So finally, I close this my presentation. Formulation man, the formula, the recipe for the success. If we successful one, whether it is a researcher or a teacher or an entrepreneur, anything, the success recipe is same. We have to have this attitude where we see like Arjun Lakshya, मतलब अर्जुन जैसा हमें बनना चाहिए कि हमें सिर्फ लक्ष्य ही देखना चाहिए, बाकी इधर उधर की चीजें हमें नहीं देखना चाहिए. Arjun was not a blind, but he had the focused inner eye. He was not seeing here and there. So this is very important to have this Arjun Lakshya, Dhruv Pratigya. We have to have this spiritual, this fervor, spiritual attitude that we should not wait to become old to know the reality of the life. The project, research project, life is a research project, and we have to attain that the big question in the life, that who am I, We need not to become the so old that we are not able to do anything. So ये हमारा attitude ध्रुव जैसा होना चाहिए कि ध्रुव ने in a very early age he asked this question and he realized the divinity and he realized self-actualization. So spirituality matters a lot because it is the bigger question, the biggest question that covers all questions. So ये द भागीरथ प्रयत्न हमारा जो एंटरप्रेन्योरशिप है, जो हमारा जुझारूपन है, जज्बा है, वो इसके आप जैसे भागीरथ जैसा होना चाहिए. जैसे गंगा जब वो लेके आए तो वी कैन ऑल्सो ब्रिंग वन मेडिसिन, द कैंसर मेडिसिन. एंड प्रहलाद भक्ति devotion is very important until unless we our ego will not be down, because it is very important. Otherwise we will become like Ravan, and this I-ness is the heightened I-ness. Yes, I have done this, I have won this award, I have won this, मैंने ये किया, मैंने ये किया. तो जो मैं करने का जो कन्वर्जन है वो कैसे होगा, जो मैं से ऊपर कैसे उठेंगे, until unless we have this attitude of surrender and to the Almighty, to the God. When we surrender to the higher power, the higher power actually takes its seat and then it walks. So this has to be understood very scientifically, that the attitude of devotion is not only a orthodoxical idea, दखलूसी कोई विचार नहीं है, but it is rather a very beneficial this concept that when we surrender to the Almighty, we go into the egolessness, and when we dissolve our ego, then higher power takes. So willpower is all about surrendering your ego to the higher self, which is not outside, which is within. So these concepts, these spiritual concepts, they are not spiritual only, they are very scientific. You can feel the difference in your own attitude, in your own manobhav. And when you change yourself, when you work on yourself, then you create the harmony outside also. You can become your magnetic, and आपको सभी लोग चाहने लगते हैं. You become attractive, you become magnetic personality. So your collaborations, joint venture, your communication becomes different, है ना. दे आपकी जल्दी दोस्ती हो जाती है, आप जल्दी एक भावनात्मक रूप से आप लोगों से जुड़ जाते हो. तो मेरा भी, मैंने ये अपना शेयर मैं करता हूँ कि जब अभी भी जैसे पीजीआई है या दूसरे भी हैं, जहाँ भी मैंने बात की तो मुझे कोई दिक्कत नहीं हुई. एवरीबडी सेज यस यस यस. I do not know, I do not host any ego again, but it happened. And I got a lot, I owe a lot to my spiritual practice, especially the heartfulness way that I could translate the research into real time product. And still I feel I am a student of spirituality, and just like I am trying to achieve the excellence while practicing this meditation. So from medicine to meditation, it was the beginning with the medicine, and now it is to end with meditation. So my take home message to each one of you is to look into this opportunity to change yourself, to work on your own mind and mindset, is precisely on your ego, intellect, and consciousness, so that you can become a better version of yourself, and that will make you a good leader, good researcher, good teacher, good student, good family man. So it's all your attitude. Thank you so much for the and this is the one poem, if you allow, if your time permits. I can play the last poem to rejoice. आकांक्षा है ये
Thank you, sir, for your inspirational journey. I request all the participants to post their queries in the chat
मेरा
box.
लाइपोसोमल
Sir,
रूपांतरण
one query from Yash. Am I audible, sir? Hello?
Hello,
With AI and automation shaping
Dr.
research,
Prashant.
how can these tools enhance the translational research in the future?
एक बार फिर से जरा बता दें, मैं मिस कर गया.
Uh, with AI and automation shaping research,
Yeah,
how can these tools can enhance the translational research in the future?
AI has a big role to play. Now we are seeing that we are having big data, and algorithm. So we can actually take help of AI and this drug target receptors and all these understanding. We can use this big data and there's a lot we are a beginner, still we have to go a long way, but this obviously this AI will play a lot of a very crucial role in research, in getting translated the research, because we have seen that the role of the QBD, quality by design, it is what the collection of these data and then developing some algorithm, so or some this validation of the optimization of this process. So of course it has a big role to play. One need to go into it, एक बार हमें उसमें अंदर गोता लगाना पड़ेगा, but AI is not everything. This is my other counterpoint, praising a lot to AI, but you can never replace human intelligence. एंड भेड़ चाल में नहीं चलना चाहिए कि हम सिर्फ एआई, अभी क्या हो रहा है कि एआई करके सबको लगता है कि एआई से सब कुछ हो जाएगा, बट एआई इज व्हाट, इट डिपेंड्स ऑन द डेटा, है ना, इट डिपेंड्स ऑन द डेटा. सो व्हेन चंद्रायन कैन फेल, है ना, बिकॉज यू आर मिसिंग द डेटा. Of course later on you found it and you made it successful. So always there is a X factor. And in the biological world, when you are facing the entire universe inside, the one cell represent the entire universe, the Brahmand. You can imagine what sort of a data available there are, and then you have to work with them. What would happen if some data are missing and you are declaring the drug useful? So this is a very complicated thing, and in my understanding, in my deep understanding, while AI will be very useful, but intuitional intelligence, the way our Charak, the way our Dhanvantri, the way our Sushrut, the Archimedes, the Aryabhat, they downloaded the information in their through their consciousness, their meditative mind. We can never be replaced. All our discoveries and the scientific breakthroughs are based on those discoveries which are not done uh, by which are not uploaded, but they are downloaded in a relaxed mind. That's what I showed the Newton. So working on the mind, working on our own mind is very, very important. Uh, One can never deny the usefulness of the machines, the machine intelligence, machine learning. ये सब तो बहुत important है. लेकिन we have to go one notch up, and we have to equally work on our mindset. We have to judge our own mindset, whether we have that the kind of that I mean कि आपके मन की क्षमता के ऊपर आपको काम करना बहुत ज़रूरी है. सो वो कैसे होगा जब तक हम अपने माइंड को रेगुलेट नहीं करेंगे. उनको एक कंसंट्रेशन नहीं कहना चाहिए, बट स्टेट ऑफ calmness, state of this tranquility. So these spiritual practices are more important than anything else. Then of course your question, I would only say there are examples. Recently IPCA Labs, I am associated with the company. They have launched one product for diabetic foot ulcer, Alcus, the namely Alcus on the Ismolol. And this was done in the one institute where they have played with this AI tool, and could create this example where this different lead of an old molecule could be produced with the help of this machine learning.
There is no more queries. So I am thankful to Dr. O.P. Katare sir for for giving the session on industry, academia, and transnational research. Sir has very nicely explained the journey of diethanol gel with current applications and formulation. With the case study, sir has explained the current therapeutic options for the treatment of psoriasis. And I hope, sir, your success recipe will be very fruitful for the budding researcher. Thank you. Thank you very much. Please kindly accept this as a token of appreciation.
Thank you so much for your
Thank you, sir.
appreciation and invitation.
Sir, okay. Sir, please be present for our validity function.
Sure, sure.
I request all the participants to stay tuned for the validity function. The feedback link will be posted in the validity function in the within the chat box.
Sir, please join the valedictory session. Link has been provided on your email, sir. And for the participants, please check the WhatsApp group. Join the link. The stream would be starting soon. Thank you.